I am Stephen G. In this demo, I'll walk you through iDEP, a web app for RNA sequence analysis. iDEP brings together powerful R packages and a large knowledge base to support visualization, differential expression, and pathway analysis, with a strong focus on quality control, exploratory analysis, and reproducibility. For our example, we'll analyze a dataset of mouse samples treated with ionizing radiation, 4 replicates per group, 8 samples total, with approximately 53,000 genes. On the iDEP homepage, select mouse as the species, choose read counts as the data type, and upload the CSV file. We recommend read counts because they carry more information and enable more accurate statistics. Let's start on the Prep tab. iDEP filters lowly expressed genes, applies appropriate transformations, and runs several sanity checks. You'll see total reads per sample and distribution plots of the normalized data. Pairwise sample relationships are available as scatter plots. On the QC tab, you'll find detailed quality metrics, including read composition by gene biotype. In this dataset, protein-coding genes account for about half the reads, while mitochondrial rRNA contributes over 20%, potentially reflecting incomplete RNA depletion. You'll also see chromosome-level summaries of total reads and expression levels. Here, one sample shows higher expression of Y-linked genes. To investigate this further, open the Marker tab. We see strong expression of the male-specific gene UTY and absence of ZIST, a female-specific non-coding RNA, in the radiation-4 sample. This suggests that this sample is likely male, while the other 7 are probably female, a detail not reported in the original study. This view also includes housekeeping genes, histone genes, and mitochondrial markers. Notably, mTRNR2, mitochondrially encoded 16S RNA, accounts for approximately 10 to 20 million reads, like 25% of the total. It is important to examine these QC plots to identify issues like this. You can plot other marker genes or any genes of interest on the Gene tab and export figures for reports or publications. Click Report to download an HTML file that collects these plots and documents your analysis settings, useful for record keeping and reproducibility. Now to the Cluster tab. Unsupervised clustering provides a high-level view of expression profiles. A heat map of the top 2,000 most variable genes shows clear radiation responses. Red means relatively higher expression. Select any cluster to zoom in and list member genes. Enable Show Enrichment to view overrepresented pathways. Here we see strong enrichment for p53 signaling among this group of selected genes induced by radiation. We also see a cluster of Y-linked genes, confirming the male sample. Switching to K-means clustering, iDEP partitions the same 2,000 genes by expression pattern into 6 groups, each with pathway enrichment summaries. You can also download an HTML file that saves settings, figures, and results. Move to the PCA tab. iDEP projects samples into interactive 2D and 3D plots. In our data, the radiation-4 sample, suspected to be male, doesn't appear as an outlier, suggesting sex differences are not driving global expression changes or responses here. However, radiation samples 1 and 2 group together, while 3 and 4 form a separate cluster. This separation, captured by PC2, echoes the clustering patterns in this cluster of genes and indicates unexplained technical variability that could confound analysis. We encourage users to explore their data using these interactive plots before running statistics. You can download all PCA results, including interactive 3D plots. For statistical analysis, open the Stats tab and compare radiation versus mock using DSCQ2. With a stringent cutoff, we detect roughly 975 upregulated and 479 downregulated genes after radiation. Use Venn or upset plots to examine overlaps across comparisons. The R-code tab provides scripts to reproduce the analysis on the filtered data, downloadable from the Prep tab. You can also download DSEQ2 results as a CSV with log 2-fold changes and adjusted p-values for all genes. Again, you can document your analysis with a report. In the DEG tab, select and investigate up or down-regulated gene sets identified in Stats. Browse annotated tables of top genes. Clicking a row shows an expression plot across samples. You can export all iDEP plots of publication quality. Additional visualizations include heat maps, volcano plots, and MA plots with optional labels. All can be saved in an HTML report. To examine enriched geoterms and pathways, here the 975 upregulated genes are enriched for p53 signaling, confirming our observation in the Clustering tab. The 479 downregulated genes are enriched for ribosome/translation processes. View results as tables, trees, or customized summaries. On the Pathway tab, run analyses such as GSEA using all genes, not just the significant genes like what we did in the DEG tab. Submit to see up and down regulated pathways, then visualize as trees, networks, or pathway heat maps. For example, you can inspect the PI53 signaling pathway and review contributing genes. Open the KEGG tab to view pathway diagrams with gene-level overlays. Red for upregulated, green for downregulated. You can also switch to single-sample GSEA to view pathway activity per sample and confirm radiation-specific patterns. Finally, the Genome tab maps expression changes along chromosomes, showing upregulated and downregulated tracks. You can zoom and inspect regional patterns in gene IDs. In some diseases, such as cancer, you can sometimes detect a clustering of differentially expressed genes on the chromosome. To wrap up, using a massive knowledge base, iDEP enables biologists to visualize RNA-Seq data and identify differentially regulated genes and pathways. It can be used at our website, just Google iDEP, or locally as an R package or a Docker container. We build iDEP so that all scientists, especially those with limited resources, can analyze RNA-Seq data by themselves. Give it a try today with our demo data.